Very good evening to all of you. We will start our discussion today as a continuation of what we studied last time. That is about the factors affecting enzyme activity. If you can remember, yesterday we talked about different factors which decides the activity of enzyme. Out of that, first one was, can anyone tell me what were the factors we discussed yesterday?
Enzyme concentration.
So it is enzyme concentration. Very good. Then
Substrate concentration.
substrate concentration.
Product concentration.
Temperature and pH.
Third one is
Effect of temperature.
but let me tell you that's not all. There are other factors. Okay. So what are these factors? We will talk now. One of them are poisons. So what are poisons? Actually, poison and radiations, different radiations are also there. These destroy the structural organization of enzyme. And I am 100% sure you know that how structural composition can be very, very important part. Without structural organization orientation, it is very difficult for enzyme to act accordingly. Next, important factors are activators. So what are activators? Activators are something which convert the inactive enzymes to the active one. Which in example I shared with you yesterday of this type of reactions. Who is going to tell me? Can you please raise your hand? Yes, Rutu.
Ma'am, yesterday you told about pepsinogens.
Great. So these are activators and things which are making inactive enzymes into active one. Last but not least, we are today going to talk about the inhibitors. So what are these inhibitors? Inhibitors, as their name itself is suggesting, is something which stops the enzyme activity. Okay, how? Let me tell you, inhibition can be of various types. What are the types of inhibition? One is competitive. Second is non-competitive. And next one is allosteric. So what are these things? What are these new things we are going to? Actually, in such cases, if you think this is the enzyme, the substrate and the competitor have similar structure. Both of them can fit into the enzyme's active site. Now, when in a solution enzyme is there, substrate is there, or I can say competitors, if you don't want to use this terminology, you can use it as inhibitor is also there. These inhibitors act as competitors. These inhibitors actually act as competitors. How? They compete with the substrate for this active site. अगर substrate की यहाँ binding हो गई, this binding is irreversible. We know that the moment product is formed, product will have low affinity towards the enzyme and it will get detached. So the binding of substrate with the enzyme is reversible. But अगर यहाँ inhibitor bind कर गया, inhibitor will never leave this enzyme. As a result, one enzyme cannot be reused. जहाँ पे inhibitor bind हो रहा है, that enzyme cannot be reused. So this is what is competitive inhibition. So in this, what happens? That inhibitors compete with the substrate. So now which factor decides कि क्या होने वाला है? Here the concentration of substrate and inhibitor decides. Because consider a situation if we have more molecules of substrate and less molecule of inhibitors. Obviously, the chances of substrates occupying the active site of enzyme will be more. But if it is vice versa, inhibitors are more in number, then obviously all inhibitors will occupy the active site, and then as it is irreversible reaction giving irreversible reaction with enzyme, वो कभी भी उससे अलग नहीं हो सकता. Understood the concept of competitive inhibitors, beta?
Yes, ma'am.
Yes, ma'am.
So now let's
मैम
yes, beta.
मैम ये इनहिबिटर्स आपने कहा कि एग्जैक्टली सबस्ट्रेट के शेप के ही होते हैं, तो फिर मैम ये कैसे बनते हैं? हाउ आर दे फॉर्म्ड?
I didn't get. How are they formed means you are talking about the chemical process.
Yes, ma'am.
I'm actually getting your question. Maybe the framing of words are a bit different. You can use how they are formed, not why they are formed. Why we require such kind of nuisance in our life when we have substrate? Am I correct? This must be your question.
Yes, ma'am. I said how only.
How नहीं बेटा, why? How means वो क्या कैसे बनता है? That is the chemical process of production of inhibitor.
Okay, ma'am.
Why means why they are there, why they are required there. See, inhibitors are not intentionally formed by body, but from the food we are taking, but from the external conditions in which we are living, there are inhibitors which are getting inside our body. Okay, so that is how they stop the enzyme activities. Understood?
Okay, ma'am.
Yes.
मैम अगर ये इनहिबिटर हमारी बॉडी में आ गया, मतलब वो एंजाइम में चला गया और अगर उसने उल्टा रिएक्शन दे दिया तो हमारी बॉडी में कुछ हार्म करेगा?
उल्टा क्या करेंगे? मतलब कैन यू रिपीट योर क्वेश्चन बेटा?
मैम अगर वो इनहिबिटर एंजाइम में चला गया तो वो उल्टा ही रिएक्शन देगा ना?
उल्टा रिएक्शन देगा
मतलब अगर सबस्ट्रेट चला जाता तो जो रिएक्शन, मतलब जो प्रोडक्ट है वो ही होता, लेकिन अगर वो इनहिबिटर चला गया तो प्रोडक्ट वो अलग होगा ना?
ऑब्वियसली प्रोडक्ट होगा ही नहीं, मैंने आपको क्या बोला? व्हाट आई सेड टू यू,
वो बाइंड हो जाएगा,
प्रोडक्ट
मतलब
तो
वो
होगा
कभी
ही नहीं।
अलग नहीं होगा
प्रोडक्ट
एंजाइम
होता
से।
तो चलो
वो
कुछ तो प्रोडक्ट
सब्सट्रैक्ट
होता,
भी होगा।
इट विल रिमेन बाउंडेड विद द एंजाइम, नो?
तो वो हमारे बॉडी में कोई हार्म करेगा?
हम तो कर ही रहा है ना भाई, आपके पास जो एंजाइम थी उसको वो इनएक्टिव कर रहा है। डोंट यू थिंक इट इज इनफ हार्म? और
Yes,
क्या
ma'am.
हार्म चाहिए हमें? अंडरस्टूड?
Yes, ma'am.
So now let's see, look into the next type of inhibition, that is called non-competitive inhibition, and how it is important. See, in case of non-competitive inhibition, substrate and inhibitors are not of same or similar structure. सेम तो एग्जैक्टली सबस्ट्रेट इनहिबिटर का नहीं होता है बेटा, बिकॉज अगर सेम होता तो फिर सबस्ट्रेट नहीं होता, इनहिबिटर नहीं होता. But in competitive, it is very easy to get rid of the problems caused by inhibitor. कैसे? If you increase the substrate concentration by any means, obviously the inhibitors will be outwitted by the substrate in competition, and that way even if they are existing in a such a low amount that they will not hamper the function of enzyme activity. But in non-competitive inhibitor, we are helpless. So what happens here in case of non-competitive inhibitor? Consider the situation if one enzyme is there. In that specific enzyme, there is a site other than the active site. So substrate will bind in the active site and inhibitor will bind into this site. The moment inhibitor is binding into this site other than active site, what will happen, beta? It will bring out changes in the active site. जैसे inhibitor is getting attached to a site other than active site, it will bring up change in the active site. So substrate will no longer be able to get attached to the active site of the enzyme. So यहाँ inhibitor को कोई फर्क नहीं पड़ रहा है कि कितने amount में substrate है नहीं है. It's least bothered about the concentration of substrate. Even if substrates are present in high amount and inhibitors are not that much, those small number of inhibitors can enter and bind into a site other than active site and can cause the binding of substrate and enzyme at stake. Understood the concept of non-competitive inhibitor, beta?
Yes, ma'am.
मैम
Yes.
ma'am.
मैम आपने बोला कि
इसमें
हमें
जो product
सबस्ट्रेट
बनेगा
का अमाउंट
One person at a time. Yes. One person at a time. Yes, Ayushi, बोलो.
मैम आपने कहा कि हमें सबस्ट्रेट के अमाउंट बढ़ा देना है तो फिर वो भी प्रॉब्लम नहीं करेगा.
बढ़ा दो ना तो प्रॉब्लम. हाँ, मतलब nothing much is actually expected, but temporarily it will be effective, no?
ओके मैम.
Okay. Now let's talk to the next type of inhibitor, that is enzyme substrate. This is in other word called as allosteric inhibitor or uncompetitive inhibitor. What happens here is once the binding of enzyme and substrate is done, then these inhibitors bind to the active site and they regulate the activity of the enzyme. Understood? They regulate the activity of enzyme. So allosteric inhibitors are not always giving you permanent inhibition. Sometimes it is observed that they are there for a good reason. Their presence control the activity of an enzyme. Okay, is it clear? Class, any doubts?
Yes. No, ma'am.
पहले श्योर हो जाओ, yes या no. So there is an excellent video which describes this in detail. I want to share that video to all of you. Yes, Khushi, your doubt?
मैम एक बार एलोस्टैटिक वापस समझाओ ना.
It's called uncompetitive inhibitor. In this case, actually the inhibition occurs in a situation when enzyme and substrate is already in a bound condition. Understood? And there are special
Okay, ma'am.
sites called allosteric sites present there.
Okay, ma'am.
Let's have a look into the video.
Okay guys, now let us talk about the enzyme inhibition. Basically, we'll be focusing on the simple stick approach of enzyme inhibition in two different slides. Here we'll be
मैम this voice is coming very low.
talking about the graphs which are helping us to understand
Actually, it is full from my side. Wait for a moment, huh?
Yes, ma'am.
Here, let's talk about tips to remember enzyme inhibition. Now, there are three types of enzyme inhibition: competitive inhibition, uncompetitive inhibition, and non-competitive inhibition. Competitive inhibition is a phenomenon in which the substrate and the inhibitor competes for the active site of the enzyme. Uncompetitive inhibition is a phenomenon in which inhibitor binds the ES complex. Non-competitive inhibition is a phenomenon in which the inhibitor can either bind active site and compete with the substrate, or it can bind the ES complex directly. To memorize the graphs of each kind of enzyme inhibition, all you need is a pencil or a pen. Now, to memorize the graph of competitive inhibition, place the pencil on the graph and hold it at 1 by Vmax value. Now, rotate the pencil in anti-clockwise direction, and this will be the graph of competitive inhibition. In competitive inhibition, the value of minus 1 by Km has changed, whereas the value of 1 by Vmax remains the same. For uncompetitive inhibition, place the pencil on the graph and push it upwards. This will be the graph of uncompetitive inhibition. Here, the value of minus 1 by Km and value of 1 by Vmax have changed. For non-competitive inhibition, hold the pencil at minus 1 by Km and rotate the pencil in anti-clockwise direction. This gives you the graph of non-competitive inhibition. In this case, the value of 1 by Vmax have changed and the value of minus 1 by Km remains the same.
We
Let's revise
discussed
this technique. For competitive inhibition, hold the pencil at 1 by Vmax and rotate in anti-clockwise direction. For uncompetitive inhibition, push the pencil in upwards direction. And for non-competitive inhibition, hold the pencil at minus 1 by Km and rotate the pencil in anti-clockwise direction.
So this is by which you can understand, through equation, you can understand how we can describe the three methods. But I still wish to know, is this part clear to you people, or you want some more explanation of these three?
Yes, ma'am.
So let me brief all of you once again. Competitive inhibition is an inhibition where substrate and inhibitors have similar structures. So they compete, they actually put a race between them. Who will first bind with enzyme? So if substrate is binding, product will be formed. If inhibitor is binding, there will be no product, that as this binding will be irreversible and it will make enzyme inactive for always. That's why competitive inhibitors जो होते हैं, they are having a irreversible bond with the enzyme. Now this situation can be overcome easily by increasing the substrate concentration rather than inhibitor. If there are too many substrate molecules available in such cases, inhibitors fall behind in competition and then substrates are occupying the active site. In case of non-competitive, here no matter how much substrate is there, inhibitor is having its own site for attachment. It is not at all targeting the site of attachment of the substrate. Understood, beta? All of you?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Now, last one here, the difference with non-competitive is this binding of inhibitor is reversible, and it can even bind with the enzyme when substrate is already bound. So that's why this inhibition can be used in a positive way as a regulator. It can regulate the enzyme action. Understood, everyone?
Yes, ma'am.
Yes, ma'am.
We will move ahead. So these are the factors basically which will basically control the activity of enzymes. Now I wish to describe a few terminologies which you people may come across when you study about different things related to enzymes. I wish to elaborate them. One of them is feedback inhibition. I told you that in case of product formation, we need to constantly remove the product. But if we have excess of product, then enzyme need to stop. So what happens is beta, there is an enzyme which will bind with the substrate to give you product. Now, if this product is in excess amount, it can directly actually bind to enzyme, and then this step is stopped, or via some means like I can say, for example, there are regulators via which they can bind with the enzyme and stop its activity. How these regulators are responsible for, Rutu, let me finish. How these regulators are responsible for enzyme stoppage of enzyme activities, that you are going to study in higher grades. But here just you need to know why we call it feedback mechanism, because whatever is getting feed, that is the product, is coming back once again to the enzyme directly or indirectly to stop the activity of enzyme. That's why this type of inhibition is known as feedback inhibition. Understood?
Yes,
मैम
Yes, Rutu. Now you ask.
मैम आपने अभी कहा कि प्रोडक्ट जो होगा वो वापस एंजाइम से बाइंड होगा.
Yes, it may. We start.
मैम पर आपने
Then it will
मैम
act
पर
as
आपने
it will act as an inhibitor or regulator, beta.
पर
There
मैम
are
आपने
options. So they will not obviously bind with the active site, beta.
पर मैम आपने वन ऑफ द लेक्चर्स में नहीं कहा था कि प्रोडक्ट का शेप बाद में मैम ऐसा हो जाएगा कि वो एंजाइम से बाइंड नहीं हो पाएगा.
Rutu, I am saying the same thing, that it will not bind with the active site of the enzyme. There may be some other site for the binding of product, ना जैसे मैंने आपको दिखाया है. What we learned is competitive inhibition the only way of inhibiting. There is non-competitive and others, no?
हाँ मैम.
वो एक्टिव साइट में बाइंड नहीं होगा, कहीं और तो बाइंड हो सकता है ना.
हाँ मैम. ओके.
Understood? चलो.
यस
I
मैम.
can see some other hands raised. If like anyone wish to ask anything.
Ma'am, can you just this whole process?
Which process?
Ma'am, feedback inhibition.
Why Ayushi? You didn't understand?
मैम बिकॉज मुझे कोई सुना ही नहीं दिया, कनेक्टिविटी इशू था ना तो.
I just described feedback inhibition procedure in which enzyme and substrate giving product itself directly or indirectly getting bound with the enzyme to stop further production of product. And as product is getting back to enzyme directly or indirectly, we call this as feedback mechanism. Understood?
Yes, ma'am.
Chalo. Now we will learn about the nomenclature of enzyme. Yes, any other doubts, Peter?
No.
So what do we mean by this word nomenclature?
Ma'am, may I?
Yes, yes.
Naming of enzymes.
Great. It means naming of enzyme. So whenever like we need to name enzyme, first of all ये question आएगा कि why we need to name enzyme? क्यों चाहिए हमें नाम enzyme का? Yes, Rutu?
Ma'am, for differentiating them according to their functions and on
it should be universally accepted by everyone. So there is a society called the International Union of Biochemists. जैसे आपने IUPAC पढ़ा होगा. So there is a society called International Union of Biochemists. वो लोग enzymes की nomenclature को they decide about the nomenclature of enzymes. Understood? So how enzymes are named? They are basically named as per their like what I can say as per their this one, the function of the enzyme, or on the substrate they are actually working. Okay. So I have a nice PPT to share with all of you regarding functions and other details of enzyme. Let me share that to you. Generally, we just mention about name of the substrate or the reaction, and we conclude it with the word ASE, that is ACE. For example, carbonic anhydrase. Anhydrase means it is going for anhydration reaction of carbonic compounds. Okay. So this is how the enzymes concept came in picture. That enzyme is sum total of N and zyme. Zyme means is cell. As we already discussed about the difference from the catalyst in a quite nice way, I'm skipping this PPT and directly coming to this part called nomenclature. So if you look into the nomenclature of enzyme, it's a very complicated procedure and not a very simple one. So तब हम enzymes के name को formally जब दिया जाता है, they have names like this. Horrible है ना? आप अगर ये name को देखो, that is EC1234. What is this? This is the name of an enzyme, but there is a meaning of this 1, 2, 3, 4. That means one is the class, that is it is coming under oxidoreductase. That means the oxidation reduction करता है. Now it can go for oxidation reduction of various compounds, but हमारी ये जो 1, 2, 3, 4 है, it is acting on aldehyde group और oxo group. इसीलिए ये क्लास के अंदर सब क्लास 2 में आता है, that is 1, 2. Then जब ये ऑक्सीडेशन रिडक्शन करता है, so who is the acceptor of electron? Here oxygen is the acceptor of electron, which is coming as subclass 3. So that is why 1, 2 and 3. And it is the fourth enzyme that is classified in this subclass. मतलब इससे पहले और 3 एंजाइम आइडेंटिफाई हो चुके हैं जो ऑक्सीडोरिडक्टेज है, जिसकी जो एल्डिहाइड या ऑक्सो ग्रुप पे काम करता है, एंड ऑक्सीजन इज द इलेक्ट्रॉन एक्सेप्टर. So I hope you understood about these implication of these words. Is this part clear?
Yes, ma'am.
तो हम देखेंगे तो हमें पता ही नहीं चलने वाला है कि भाई कौन से एंजाइम के बारे में बात कर रहा है. बट इफ यू हैव द इंडेक्स of all these classes and subclasses, you can easily find it out. Actually scientists do it in that. Now let's talk about the classification of enzymes, how we can classify enzyme into different types depending on their function. So first one is oxidoreductase. As I told you, they go for oxidation reduction reaction, catalysis. They catalyze the oxidation reduction reactions. Then is transferase. They transfer the group. Now how they transfer the group? Here beautiful examples are there. EC1 oxidoreductases. You can see that A is there, B is there. Now A is giving some kind of electron to B. So A is getting oxidized and B is getting reduced. Understood? I explained it in terms of electron, but in lower grades you studied the concept of oxidation reduction in terms of donation and acceptance of oxygen and hydrogen. You can think it like that also. Any reaction you take, oxidizing and reducing agents are there and opposite is formed. This part is clear to all of you? Think and let me know.
मैम
Yes, beta.
मैम वो वाला पोर्शन आप फिर से एक्सप्लेन करो ना मैम. वो ए के साथ वो ऑक्सीडेशन कैसे हो गया और बी के साथ रिडक्शन.
You studied oxidation reduction reaction in grade 10.
यस मैम.
Then what is the confusion? Consider करो ये reducing agent है. Consider this is the reducing agent and this is the oxidizing agent. तो reducing agent खुद क्या होता है? Oxidize होता है. So what it will do to get oxidized? Oxidation मतलब either, what do we mean by oxidation?
Removal of H, hydrogen and addition of oxygen.
बायो टीचर से केमिस्ट्री वाली टीचर की काम करवा ही लोगे, बट आई डोंट माइंड बिकॉज दीज टू सब्जेक्ट्स गो हैंड इन हैंड, है ना. चलो ए रिड्यूसिंग एजेंट मतलब वो ऑक्सीडाइज होगा. अब ऑक्सीडाइज होने के लिए उसको क्या करनी पड़ेगी? इट कैन डोनेट हाइड्रोजन हियर.
Yes ma'am.
Form of B. Done.
Ma'am, got it.
Got it? Great.
Yes ma'am.
Chalo. Now we will move ahead with the next type of enzyme. Here they actually mentioned about the reaction. Here H2O2, H2O2. So H2O is formed. It is getting oxidized, reduced. You can take example of this also. Is this example clear to all of you?
Yes, ma'am.
Yes, ma'am.
Now the transferases. What do we mean by transferases? Transferase means they are associated with some kind of transfer. So what they do? Basically they transfer a part into other molecule. For example, this B part present here is getting transferred into C. That is how it goes for transferase reaction. Here ATP, adenosine triphosphate, is giving one phosphate to the hexose. So it is giving one phosphate to the hexose. This is example of transferase reaction. Clear, beta?
Yes, ma'am.
Now, next example, hydrolysis. You know what is hydrolysis. I told you, isn't it? Hydrolases are different from hydrolysis. Hydrolases does what? They actually different mean to say, for example, A and B is having a bond. This bond is broken down similar to the type of hydrolysis reaction I described in case of this one, carbohydrate. This H2O will be divided into H and OH minus, and they will bind with A and B. So as the valency of A and B is satisfied, the bond between A and B will be broken. So basically they are hydrolases, that means they go for lysis or breakdown of the bond. But I mean to say that spelling is different over here. Hydrolases, na ke lysis. So do not get confused between these two terminologies. Okay? Hydrolysis is the reaction and hydrolases are the group of enzymes which catalyze the reaction. Is it clear?
Yes.
Now lysis. Lysis actually simply break down the bond. They go for lysis. In this case, no addition of H2O is not added, but still the bond is broken down. So such type of enzymes who catalyze this kind of reaction is called lyase. Next is isomerase. This is so very easy for you to understand. For example, you can see the glucose is converted into fructose, aldose is converted into ketose. So isomerase go for isomerization reaction. Next is ligase. The way ligases break down bond, ligase create bond between two substances. For example, this A and B were separate. Okay. Using ATP, there is formation of a bond between A and B. This is what is the function of ligase. And last but not least is the translocase. Yes.
मैम ये लाइगेस वाला फिर से आप एक बार रिपीट करो ना.
What is the part that you didn't understand, beta?
मैम अभी आपने बोला ना कि लाइगेस कन्वर्ट्स इनटू वो वाला कुछ.
They create a bond between A and B. Understood?
ओके मैम.
मैम
Yes, beta.
मैम ये जो ए में जो हाइफन करके बी लिखा है मैम, वो बॉन्ड है या फिर माइनस की साइन है?
बॉन्ड है बेटा. इट्स बॉन्ड.
ओके.
केमिस्ट्री में माइनस साइन कहाँ से आएगा? यहाँ नहीं आएगा. ऑब्वियसली इन फिजिकल केमिस्ट्री देयर में भी माइनस. ये जितने भी है ऑल ऑफ देम आर बॉन्ड्स. यू विल अंडरस्टैंड. अच्छा, आपको मैंने वो गुड न्यूज दी थी कि ये चैप्टर आप अच्छे से पढ़ लो तो क्या होने वाला है आपके साथ. You remember I gave you one good news. This same chapter, almost 90% of this same chapter is there in grade 12 chemistry.
यस मैम आपने
ये
बोला.
पढ़ लो तो तब free. That's why I am stressing so much on this. Now, last but not least is translocase. Do not get confused with the translocase and transferase. Transferase is responsible for shifting group from one substance to another substance. whereas translocase is responsible for shifting atoms and molecules or the functional group from one position of a same substrate to the other position of the substrate. Understood? जैसे कि समझो, let me tell you, जैसे हम लिखते हैं ना CHO, so if like I am giving to you just example that if it is an isomerization reaction, obviously, if like one of these H is going over here and this one is also getting over here, you can see that if these two H are moving to the first carbon atom, there will be formation of a new substance. Or let me give you another example. Consider there is a compound like this, where chlorine is present in the second carbon atom. The role of translocase is to send it to the, rather to say, to send it to the this carbon atom. So obviously this type of changes are done by the translocase. Understood? The difference between, yes, beta.
मैम क्या आप यही एग्जाम्पल ट्रांसफरेंस में कैसे होगा वो बता सकते हैं?
ट्रांसफरेंस में दो अलग कंपाउंड होंगे बेटा, समझो इसके अलावा एक कंपाउंड होगा ऐसा, so in that chlorine is going from this compound to that compound.
Okay ma'am.
Understood?
Yes ma'am.
So that is all about classification of enzyme, and with this I am done with the topic of enzyme, and I just want to share like one nice video of enzyme with all of you as a revision video. Every time we complete one part, we go for revision. I just want to do that only. Can I?
Yes ma'am. Yes ma'am.
It's like, you know how what I feel. जब हम खाना ज्यादा खा लेते हैं ना, देखना थोड़ा ऐसे हिलडुल लेते हैं तो क्या होता है, खाना सेटल डाउन थोड़ा हो जाता है, थोड़ा हमें अच्छा लगता है. सिमिलरली हम बहुत सारा पढ़ लेते हैं ना, एक बार उसकी रिकप हम देख लेते हैं तो फिर it will be very easy for us to understand that part, and it should be in a funny way. That's why. Oh, sound I need to.
Should we care about enzymes? Well, enzymes are really important in
Is the screen visible and sound is audible to you people, beta?
Yes, ma'am. It is perfect.
Yes ma'am.
Great, great.
Yes ma'am.
This is a video about enzymes. Why should we care about enzymes? Well, enzymes are really important in biology because they act as catalysts, which mean that they make things happen faster. And faster is better. Imagine if you had a choice of 2 cars to take to school or work each day. Which would you choose? This one or this one? I think you would choose the Lamborghini because faster is better. Well, just like that Lamborghini, enzymes make chemical reactions in the cell go faster. If we didn't have enzymes, your body wouldn't be able to digest that cheeseburger you just had for dinner last night, or produce the energy you needed to win that marathon you were in last weekend. In fact, we simply could not survive without enzymes because none of our chemical reactions in our cells would happen fast enough. So enzymes are really very important. Enzymes are proteins, and like all proteins, they are made up of amino acids. The sequence of those amino acids dictates how the enzyme will fold and what three-dimensional shape or structure it will have. And the shape or structure for an enzyme is crucial for its function, because enzymes are able to speed up chemical reactions by physically interacting with the reactant or reactants of a chemical reaction. The reactant that an enzyme acts upon is called its substrate. An enzyme interacts with its substrate at a specific location called the active site of the enzyme. The shape of the active site is complementary to its substrate, like a key to a lock or a dancer's foot to her shoe. This complementarity makes an enzyme specific to its substrate, meaning the enzyme can only catalyze the reaction for its specific substrate. The interaction of the substrate and enzyme leads to the formation of an enzyme-substrate complex. When a substrate interacts with the enzyme at the active site, the shape of the active site changes slightly to accommodate the substrate due to chemical interactions between the substrate and active site. This shape change allows the substrate to fit more tightly into the active site and is referred to as the induced fit model of enzyme-substrate interaction. Since enzymes are proteins, they are sensitive to the same environmental changes that affect protein folding, such as temperature or pH. All enzymes have an optimal range of temperature and pH at which they work best. Fluctuations from the optimal range can lead to enzyme denaturation. Denaturation alters the shape of an enzyme, including its active site, which can prevent the enzyme from being able to interact with its substrate. Enzyme function can also be blocked by inhibitors. Enzyme inhibition is extremely important in health and medicine, since most medications that you can buy from a pharmacy or that are prescribed by your doctor function as inhibitors of enzymes in our bodies. Enzyme inhibition falls into two broad categories: competitive inhibition and non-competitive inhibition. Competitive inhibitors resemble the normal substrate for the enzyme and bind at the active site of the enzyme. They therefore compete with the substrate for occupancy of the active site. An example of a competitive inhibitor is a family of drugs known as statins, such as Crestor and Lipitor, that are used to lower cholesterol levels. Statins function by competitively inhibiting an enzyme in the body that is required for the synthesis of cholesterol. Non-competitive inhibitors, on the other hand, do not resemble the substrate and do not bind at the active site. They bind the enzyme at an alternate location, but the binding of a non-competitive inhibitor alters the shape of the active site, preventing the substrate from being able to bind. An example of a non-competitive inhibitor is the highly toxic poison cyanide. Cyanide is so lethal because it blocks the function of an important enzyme needed for cellular respiration. Without cellular respiration, our cells die because they cannot produce the ATP needed to carry out their normal functions. So in conclusion, enzymes are really important proteins that function by speeding up the chemical reactions in our bodies. They interact with the substrates at their active site, forming an enzyme-substrate complex. A large part of the world's pharmaceutical industry relies on the fact that enzymes can be inhibited. This inhibition falls into two broad categories: competitive inhibition, where the inhibitor competes with the substrate for binding to the active site, and non-competitive inhibition, where the inhibitor blocks enzyme function by binding at a site other than the active site.
So I think it was a nice revision class.
Yes, ma'am.
Okay.
Yes, ma'am.
We will now talk about a molecule which is very small in size and as compared with the big molecules we studied, like proteins and carbohydrates and lipids. But its size doesn't mean that it is insignificant. It is one of the most significant substance which is in association with all types of biomolecules and control its activity. Okay, so we are going to talk about the energy currency of the cell. Who is going to tell me what is the energy currency of the cell?
May I?
Yes,
ATP,
so
adenosine triphosphate.
you may ask, ma'am, इसमें क्या खास बात है? What you are going to tell me extra? I know what is energy currency of the cell, but today we are going to learn about the structure and how it stores actually the energy that we are going to learn. Okay, so this is the structure of ATP, that is adenosine triphosphate. You can see this is the adenosine, ribose sugar may be there, and 3 phosphates are attached here. Now, adenosine can withstand attachment of 2 phosphates very easily. It can actually accommodate the first phosphate happily, second phosphate also happily. But for binding of this phosphate and oxygen in case of the third one is not at all possible in normal situation. Why? Because see, phosphorus is also electronegative and oxygen is also electronegative. So this binding require a lot of energy, and it can be done only in presence of energy. So what actually ATP do? It moves here and there in the form of ADP, that is adenosine diphosphate. And apart from that, PI is also present along with it. PI means inorganic phosphate, that is inorganic form of phosphate. Wherever they catch hold of any amount of energy, they utilize that energy to make this third third phosphate group's attachment possible and get converted into ATP. But ATP is useless if it cannot get back into the form of ADP. उसको ADP फॉर्म में जाना है तो पड़ेगा ना. जैसे आप पावर बैंक्स यूज करते हो ना आजकल, तो एवरीवन इज यूजिंग, इज इट? यस और नो?
Yes, ma'am.
हाउ पावर बैंक वर्क? क्या करता है पावर बैंक?
And it charges.
Ma'am, it stores energy.
वो एनर्जी को स्टोर कर लेता है. जहां आपके लैपटॉप के पास चार्जर अवेलेबल नहीं है, इट कैन ट्रांसमिट द एनर्जी, इजंट इट? सो सेम इज डन हियर. वो क्या कर रहा है? एटीपी स्टोर करके रख रहा है. बट नाउ इन प्ले in presence of ATPase enzyme, these are special type of lysases which break down this bond. जैसे ही ATPase enzyme will be there, ATP will be broken down in the form of ADP and PI. So the energy released will be utilized by biological substances. That's why we call that ATP is the energy currency of the cell. This part is understood.
Yes.
So with this, I am like done with today's topics.
Ma'am,
Yes, yes, beta.
ma'am, will you please repeat the last 2 sentences which you told about ATP?
Like it's very difficult to remember what are the last 2 sentences I spoke. But anyways, can anyone guide me? Which part, up to which part you heard, beta?
मैम वो एक्चुअली मेरी अचानक लिंक टूट गई मैम, क्योंकि यहाँ पे मैं जिस रूम में बैठी हूँ वहाँ पे कुछ कॉन्वर्सेशन चल रही थी.
That's why I am saying कि up to what part you heard?
मैम ATPase enzyme तक सुना.
So ATPase
special type of
enzyme will catalyze the reaction in which ATP can be broken down into ADP and PI. So as this bond is broken, so obviously the energy that was associated for bond formation will be released and which can be used by others. That's why ATP is known as the energy currency of the cell.
Okay, ma'am. Thank you.
Wow. Anything else if anyone wish to ask? Because tomorrow we are going to finish this chapter. Tomorrow we will study about nucleic acids and then we are done.
Ma'am,
Yes, beta.
galactose is called brain sugar.
Yes,
Ma'am,
galactose is
brain
called.
sugar.
In
हाँ,
brain,
ma'am, brain sugar.
it's present. It is detected in brain. That's why.
Okay.
Anything else? Anyone wish to ask something, beta?
No, ma'am.
No.
Nothing. So up to whatever
Ma'am?
it is. Yes, yes, beta.
मैम वो जो आपने लास्ट वाला समझा है उसमें आपने एक रिएक्शन लिखी थी ADP प्लस PI, तो उसमें PI मतलब इनऑर्गेनिक फॉस्फेट बोला ना आपने?
Yes, yes, beta.
ओके मैम.
For example, let me tell you why I wrote inorganic phosphate. Because in biomolecules as we study that phosphate can be present in phospholipids also. Isn't it? That phosphate is known as organic phosphate or phosphoprotein also. Inorganic phosphate means basically H3PO3 or H3PO4.
ओके मैम.
Anything else? Okay, then let's close our lecture over here. Tomorrow we will conclude our chapter with the last part that is left, that is DNA and RNA, that is nucleic acid. Okay, so bye to all of you. Very good evening. Bye.
Ma'am, thank you, ma'am.